salvage pathway. Right? There are two mechanisms. One is the de novo. It's a newly synthesizing purine rings. So salvage pathway means you are utilizing them again, means by recycling. Okay. So here in the purine, I mean metabolism also uh, similar to the pyrimidine, or pyrimidine is similar to the purine metabolism. Okay. But just like the purine metabolism needs a lot of amino acids, carbon dioxide, right? Then ammonia, glutamine. We need a lot of things. But here, as the ring is here very simple because it is a single ring. Okay, so it contains simple molecules, not the so many compounds. Not so many compounds. Here we need just only few compounds only. So what are those few compounds here? Aspartate, glutamine and CO2. Okay, aspartate, glutamine and CO2. So these three contribute uh, the formation of the pyrimidine ring. Okay, and one more difference here in the purine synthesis. Usually, what happens? A ring formed on the ribose sugar. But here in the pyrimidine synthesis, the ring formed first, and then it attaches to the ribose sugar. Okay, so that is a main difference. Okay, between the purine synthesis and pyrimidine synthesis. So already we discussed yesterday, right? The ring forms on the ribose sugar, means the two rings. Okay, on the ribose sugar. But here the ring forms first and then attaches to the ribose sugar. So that is a difference here. Okay, then this is a pyrimidine structure. I mean, here, as I told you, it is derived from the simple molecules, like it is having glutamine amide, aspartate, then bicarbonate. Okay, so it is having only these three. Okay, what are these three? Just it need a glutamine, carbon dioxide, and a ATP. Okay, so aspartate. So this is a pyrimidine nucleus. Okay, it's a single ring structure. Now, coming to the biosynthesis. So, how this is synthesized. So, before this, it has to synthesize three end products, right? So, what are the three end products? Yesterday we discussed adenine monophosphate, adenine diphosphate, adenine triphosphate. Then guanine monophosphate, diphosphate, triphosphate. That is the RBC. I mean the ribose, right? Then coming to the deoxyribose, it has to convert deoxy deoxyadenosine, then deoxyguanosine or guanine, monophosphate, diphosphate, triphosphate, like that, right? But here, what are the end products here? Uridine monophosphate, cytidine monophosphate, then thymidine monophosphate. Okay, so if it is a cytidine monophosphate, thymidine monophosphate, uridine monophosphate. These are the end products because here we are not talking about a specific RNA or DNA, right? So we are talking about the nucleotides. Nucleotides. So the nucleotides, I mean, they combine to form either a RNA structure or DNA structure. It's nothing but a strand, right? Either RNA strand or a DNA strand. But here we need three, right? Uridine monophosphate, cytidine monophosphate, and thymidine monophosphate. These three are important. Okay. Then where the synthesis of pyrimidine occurs, the same thing, right? Cytoplasm. Mainly it occurs in the liver cells or other rapidly dividing cells. Okay. Now coming to the steps. So initially we need the glutamine, carbon dioxide, then ATP. These three. So these three, okay, will yield carbamoyl phosphate. Okay, because it is having the carbon dioxide, glutamine, ATP. These three are combined together to give a carbamoyl phosphate. So this is first step. Okay. Then in the second step, this carbamoyl phosphate along with this aspartic acid or aspartate is converted to carbamoyl aspartate, carbamoyl aspartic acid or carbamoyl aspartate. Okay, initially in the first step, carbon dioxide, glutamine, ATP, there is three. Okay, with an enzyme, carbamoyl phosphate synthase 2, it converted to the carbamoyl phosphate. This is the first step, right? Then after combining with aspartic acid, it's converted to the carbamoyl aspartate or aspartic acid. Okay, here also we need an enzyme called aspartate transcarbamylase, right? Then this is the second one. Then coming to the third one. Third one means this carboxyl aspartic acid, okay, undergoes cyclization. Okay, it undergoes the cyclization so that here the complete ring get closed. So this is the same one. It undergoes the cyclization. Okay. So what this cyclization do? It converts this into dihydroorotate. After cyclization, the ring closed. So this is single ring formed, right? So this is called the dihydroorotic acid or dihydroorotate. Okay. Then this dihydroorotate is oxidized. This is oxidized. Then after oxidizing, it is converted to orotic acid. Initially dihydroorotic acid, then after oxidation, it will be converted to the orotic acid. Okay. So yeah, this is the orotic acid. Then after this orotic acid formation, it combines with the I mean PRPP. You know this one, right? Phosphoribosyl transferase. So orotate phosphoribosyltransferase transferase with the help of this enzyme, okay, along with this PRPP, this orotate will be converted to the OMP. What is an OMP? Orotidine monophosphate. Orotidine monophosphate. Okay. Here it needs an enzyme, that is, what is that enzyme? Orotate phosphoribosyltransferase transferase. Then this OMP, orotate monophosphate, will be converted to UMP. Okay, UMP. What is an UMP here? UMP means uridine monophosphate. Okay, using this orotylic acid decarboxylase, this OMP is converted to the UMP. Okay, then this UMP, okay, it's a monophosphate, right? It's a monophosphate. This monophosphate is converted to diphosphate, then triphosphate. Okay, so uridine monophosphate, diphosphate, then triphosphate. Okay, here because of this phosphorylation means adding the phosphate, right? So UMP will become from OMP it converts to UMP, then from UMP UDP, then UTP. Then from this UTP it converted to CTP, cytidine triphosphate. Okay, so here how many pyrimidines created, how many nucleotides got created? UMP1, UDP, UTP. Okay, these three. And then from the UTP, it creates the CTP, cytidine triphosphate. Okay. Then we need one more. Cytidine is finished, then uridine finished. Then we have the thymidine. So how the thymidine is converted? So thymidine means here the UDP, it is converted to deoxy-UDP. Okay, deoxyuridine diphosphate. So it is the DNA, right? This is the RNA. UDP means RNA. Deoxyuridine diphosphate. Then this will be converted to a monophosphate, and then this UMP, I mean deoxy UMP will be converted to the TMP. So thymidine monophosphate, right? So we have three: thymidine monophosphate, cytidine triphosphate, then what is the other one? Uridine, cytidine, then thymidine, right? So these three are formed. Clear? So this is how the pyrimidine synthesis will occur. Okay. So how many steps here? Totally we have 12 steps. So within these 12 steps, the UMP, UDP, UTP, okay, CTP, then deoxy UDP, then UMP and TMP also got created. Okay. So these are the steps in the pyrimidine biosynthesis. Now you can see here the same carbamyl phosphates synthase 2 acts on the carbon dioxide plus glutamine. Then carbamyl phosphate is the first step. Then carbamyl aspartate. After adding the aspartate, it will be converted to the carbamyl aspartate. Later, dihydroorotate. Okay, cyclation will happen. Then orotate. After removing the or after reduction, it will be converted to the orotate. Sorry, oxidation. After oxidation, will be converted to the orotate. Then orotate will convert to orotate monophosphate. Then uridine monophosphate. Then this uridine monophosphate will be, you know about this one, right? We yesterday also we discussed thiouredoxine. It converts to deoxy. Okay, then this UDP will be converted to UDP, then CTP. Here this converted to DUMP, DTMP. So cytidine, then DTMP. Okay, so this is the biosynthesis of the pyrimidine. Then catabolism, how the breakdown of the pyrimidines happens. So mainly the breakdown happens in the liver. Uracil, okay, uracil will be converted to the dihydrouracil, then beta iridopropionate, then it will be diffused into alanine NH3CO2. Okay, then this methyl cytosine also converted to thymine, then dihydrothymine, then it will be breakdown into beta amino isobutyrate NH2 plus CO2. So this is a pathway, the breakdown pathway of the pyrimidine or catabolism of pyrimidines. Okay, then inhibitors are there. So what are the inhibitors? Inhibitors means some drugs are there. Okay, some drugs are there. They will inhibit the formation of the purine as well as the pyrimidine. So these inhibitors is not only for the pyrimidine we're talking about. These are the inhibitors of the purine also. Okay, so there is one drug called the sulfonamide. They inhibit the reactions of purine nucleotide synthesis requiring folic acid. Okay, so some purine nucleotides need folic acid. You know the PABA, right? Paramino benzoic acid. Okay, they need the folic acid. So, I mean, for the synthesis of the purine. Okay, so this sulfonamide they inhibit the synthesis of purine nucleotides where there is a need of the folic acid. Okay, used as bacteriostatic drugs to control bacterial infection. So this sulfonamide we use to treat bacterial infection by inhibiting the purine nucleotide synthesis. Okay. Then methotrexate, aminopterin, then trimethoprim. They inhibit the reaction requiring folic acid for purine nucleotide synthesis. They are also acting as the same. Okay. Used in the treatment of cancers like leukemia, then used in the treatment of bacterial infection, UTA, the same mechanism. Then there is one more called the mercaptopurine. It is a structural like a purine basis. Okay, it is converted to 6-thioinosine monophosphate by the enzyme HGPRT called lethal synthesis. Then thioguanine also there. Okay, we have different types of inhibitors. Okay, agesterine, then colchicine, then probenicid, allopurinol. Okay, so these are the different inhibitors. Okay, again the same, okay, but given in the form of table. Clear. So this is about the pyrimidine metabolism. Clear. Then if you talk about the disorders of the pyrimidine metabolism, okay, so usually the deficiency leads to megaloblastic anemia. Megaloblastic anemia. Then growth retardation also will be there when there is a pyrimidine nucleotide metabolism ceases. Okay, so in that cases we use the uridine administration, and we spoke yesterday about the severe combined immunodeficiency, right? SCID, bubble baby. Okay, even that is also happens when there is a deformity in the pyrimidine nucleotide formation. Okay, yeah. So this is about the nucleotide metabolism. So this is very, very important one. Then definitely you will be getting a long answer question. Okay, and short answer questions also in the paper 2 of biochemistry. Okay, so don't ignore this one, because definitely they will ask a question on gout also, primary gout and secondary gout. Okay, and at least salvage pathways they ask. Okay, if they don't ask the de novo also, at least they will ask the salvage pathways. Okay, of pyrimidine also they may ask, but pyrimidine they may ask the short question. Okay, yeah. So this is very, very important topic, right? Okay, so this is about the pyrimidine and purine metabolism. Now let us see the other topic called the replication of the DNA. I think most of you are aware of this replication, right? So anyway, DNA replication in eukaryotes and prokaryotes. So this is one of the interesting topic, and also an important for examination also. Sometimes you may get the long answer question also on this. Okay. So what is the DNA replication? So yesterday we discussed about the central dogma. Central dogma means from DNA to DNA. The formation of one DNA or the copy of DNA from the original is called a DNA replication. See, one cell is there. Okay. So the cell want to multiply or duplicate. Okay. So not only the cell body, it has to distribute the DNA component also. Then definitely it has to make a copy of DNA. Okay. Then that copied DNA will be moved to the, the offspring or the other cell. Got it? So here, what is the purpose of replication? The process by which DNA is essentially doubled. Got it? So that is happening within the dividing cell, not the cell, which is not undergoing the division. Okay, if there is any cell division, then only the DNA replication will happen. Okay, so but here the DNA replication is almost similar in the eukaryotes and prokaryotes also, but the slight difference is there between the enzymes. Okay, and also between the use of some proteins. Okay, so slight difference is there between the eukaryotes and prokaryotes. I will tell the differences also, because usually the prokaryotic DNA is a round, right? And eukaryotic DNA is not round, linear. Okay, so there is some difference. Okay, we'll see the difference later. So first, let us see what is the DNA. The process by which DNA is essentially doubled. It is an important process that takes place within the dividing cells. So the original is converted to two. Okay, so these are the copies. Can you see? This is a double strand one. It unwinds, then it creates a new strands on the one older strand. Then again on the older strand, it creates a new strand. Now these two, okay, these two original, and from the original, these two copies were formed. But in these two copies, one is the old strand. In the second one also, one is the old strand, and new strand, complementary strand got created. Okay, so this is the method of replication. Okay, so usually we call this as the semi-conservative. What is a semi-conservative? Semi-conservative means usually what happens, uh, this will enter into one cell and this will enter into one cell. Means the newly formed cell contains one old strand of DNA, the other one is a new strand of DNA. The other cell also having the same, right? That is called semi-conservative. Okay, let us see what is a conservative. See, conservative means the two strands enter into one cell, the other cell will receive two new two new strands only. Okay, so this is called the conservative, but this is not happening. This is not happening, right? So usually what happens in the first generation, in the first replication, so if it is a conservative mode, these two will enter into the new one. Okay, or cell is divided into two cells, right? So one cell will reach, one cell will get the two old, okay, the other cell will get the two new, but this is not happening. Okay, so it's happening semi-conservative. Semi-conservative means one is the old one is the new one. Okay, here the one is old one and it's a new one. Okay, then again in the next division, then it gets the old one. Okay, it gets the old one. Okay, from here, and it creates a new one. Then this is already newly created, right? Again, this newly created will generate one more complementary strand. Okay, so these are the new only. Okay, so out of four here, in the second division, I was talking about the second division. Okay, F2 division. So out of four, two are having two old strands, then the two are having the complete new strands. This is a semi-conservative. Then this is the dispersive. Dispersive means having both. Okay, having both like this. So the dispersive is not correct and conservative also it's not correct. Semi-conservative. So how can you consider the semi-conservative replication is happening means uh, actually Michelson and Stahl. So these are the scientists. So they made an experiment. Okay, uh, by using some density gradient centrifugation, and also they made two nitrogen isotopes. Okay, means they added some isotopes to the nucleus. Okay, and they waited till the cell division happens, and then they extracted the DNA and they checked, okay, whether the cell is having the new strand or old strand. So they conducted an experiment. Okay. So with that experiment, it got proved that the DNA replication is happening in a semi-conservative method only, not the conservative method and dispersive method. So no need to write that experiment, just mention the names. So as per the Michelson and Stahl experiment, it is proven that the DNA replication is happening in a semi-conservative method. Okay, that is enough. Then coming to the replication. So DNA replication in prokaryotes. First, we will see how the DNA replicates in the prokaryotes. What do you mean by replication here? Replication is nothing but making two copies, right? One copy is converted to two copies. Replica, it is creating a new one, right? So how this DNA replication is happening, let us see. So what do we need to replicate the DNA? First of all, we need a template, right? We need one copy. See, DNA is there. DNA is a double stranded helical structure, right? So that double strand, it has to get unwind. It should be splitted to two separate strands, two separate single strands. So one strand is called a DNA template. So we need a DNA template. Later, building blocks of DNA. We have to build one more strand complementary to the DNA template, right? So one more complementary strand means what we need? We need the nucleotides, right? We need the nucleotides. Okay, without nucleotides, how can you build the complementary strand? So it's not possible. First of all, we need a DNA template. Then complementary to the DNA template, we need the we need the nucleotides. So those nucleotides are nothing but the building block of the DNA. Right? Yesterday we discussed about the nucleotides, right? So those nucleotides are important because when you join all the nucleotides, then it will become a DNA complementary strand, right? So what type of DNA building blocks we need, we'll discuss. Then later, replication also needs some proteins and enzymes, endonucleases, exonucleases, primers, helices. So we need a lot of proteins and enzymes. And later we need a primer also. Primer means initiation, right? So there is a primer. So from the primer only, the replication process will start. Okay. So we need these things to create a new DNA. Okay, let's start. First we will see the DNA template. So DNA template is it is a double stranded, double stranded. Okay, double stranded, uncoiled. Okay. So you know this one. So one strand runs in 5-3 direction, the other strand runs in the 3-5 direction. So both are coming in a opposite direction, right? And these are complementary. Yesterday we discussed AT, TA, GC, GC, or AT, CG like that. Okay, so complementary. So we need one strand. Yes, these are double stranded, right? So to replicate this coiled strand, this double strand should be unwinded to a single strand, right? So this is called a DNA template. We need a DNA template. Okay. Then on this template, one more template will be created. Then on the second template, one more complementary template will be created. Okay. Then these two get base paired and these two get also base paired. So that one DNA template will be converted to the two DNA templates. That's nothing but a replication. Got it? So here, after unwinding the DNA template, each strand serves as a template for the replication. Remember complementary base pairing. You know this one. Okay. Then what is the second one? Building blocks of DNA. What is the building blocks of DNA? DNTP. What is the DNTP? Dehydroxynucleotides. Okay. One is dehydroxy adenine triphosphate, then thymine triphosphate, then guanine, then cytosine. We need all the four. Why? See, this is a nucleotide, right? So if you combine the nucleotides, then it will be converted to a strand, right? If there is no nucleotide, then how will you build the complementary strand? It's not possible. So we need these NTPs, right? Deoxyribonucleoside triphosphates are nucleotides we need. Okay. Then why we need the triphosphates only? Always adenine triphosphate, okay, guanine triphosphate. Why? Because one nucleotide will be connected to the other nucleotide using the phosphodiester bonds only, right? Phosphodiester bonds only. So we need the more phosphate. Okay, after connecting with this phosphodiester bond, the remaining uh, phosphates will be liberated. Okay, so we need energy to create the or to replicate. We need the energy. Okay, then these are the builders. We need some enzymes and proteins also, right? So what we need? We need a DNA polymerase enzyme 1,3. We need a helicase and actual DNA A, B, C. DNA A is a initiation protein. DNA B is a helicase. DNA C is a helicase loader. Then we also need the single strand DNA binding proteins, gyrase and ligase. Okay, just to remember the symbols. So we'll be discussing all this one by one. Okay. Later we need a primer. Primer means here it will start. Here it will start the formation of a new strand. Okay. In order for the DNA copying machine to work and add nucleotides, a 3 OH 3 prime OH needs to be able to form a phosphodiester bond. Okay. We need the this primer. Then from this primer only the initiation will start. Okay. Then coming to the, I mean, we are talking about the prokaryotic. We are talking about the prokaryotic. Later we'll be discussing the eukaryotic. Then coming to the DNA polymerases. So what are the DNA polymerases here? See, one is the strand. We need a template, single strand. Later we need the building block. Building block means uh, adenine triphosphate, guanine triphosphate, deoxy. Okay, we need the nucleotides. Later we need the proteins and enzymes. Then primer. Primer is okay. Then coming to the builders. In the builders, we have different type of proteins, right? So here we are talking about the enzyme called the DNA polymerase. So what the DNA polymerase do? Actually, DNA polymerase are three types here. One is polymerase 1, polymerase 2, and polymerase 3. Okay. So what is polymerase? It will do the polymerization. Polymerization means it will connect connect the nucleotides with the phosphodiester bonds, right? So we have three polymerases. So polymerase 1, it will do the exonuclease activity, removes primers and replaces them with the DNA. Initially, see the primary attached, right? So this polymerase 1, what it will do, once the formation of the a new strand happens, then it will remove the primers and it will add the nucleotides in the place of this primer. Okay, that is the polymerase 1. Then what the polymerase 2 do? It will do the repair function. Okay, when there is any missing proofreading, we call it the proofreading. Proofreading means when it is reading the strand, when something is missing, then it will do the repair. That's called the polymerase 2. Then polymerase 3 means it will keep on adding the nucleotides onto the complementary chain. Okay, onto the chain. So in which direction? 5-3 direction. 5 prime, 3 prime direction. Always the replication happens in 5-3 direction only. Okay, so these are the three polymerase enzymes that are helped in the synthesis of the new strand. Okay, what it will do? It will do the polymerization, adding the nucleotides or combining the nucleotides onto the complementary base pairs. Okay. Then coming to the DNA polymerase, okay, let us see what is the exonuclease and endonuclease. Exonuclease means it will cut at the end, exo. Endo means it will cut in the between, in between. Yeah, if you have any doubts, you just ping me, okay? Because usually it's happening like one side only, okay? So if you have any doubts, you just ping me, okay? You can even discuss also with me because we have that option of, unmute your mic. Okay, so yeah, I hope you are getting. Yeah, yeah, just a moment, please. Just to give a moment. I'm back. So hope you are online. Okay. So what is this nucleus? Nucleus means it will digest the nucleotides, right? Exonucleus means it will cut at the ends. Endonucleus means it will cut in between. Okay. So these are exo and endonucleus definitions. Then, as we are talking about the DNA polymerase, right? DNA polymerase 1, 2, 3. Okay. So usually what happens? The DNA polymerase adds the nucleotides in this direction, 5'3" direction, right? See, 5'3" direction. DNA polymerase, it will keep on adding the nucleotides in the 5 prime, 3 prime direction, right? So, see, it is keep on, this is a DNA polymerase. So, it is keep on adding the nucleotides in this direction, okay? I mean, this is a normal one. But sometimes what happens on this way, I mean, in this way, for example, here there is a and T are there. A and T are there. We don't need this A and T. Then it will cut this A and T. Okay, it will cut an A and T, means it will try to remove the A and T. Got it? It will try to remove the A and T also. Got it? And in the place of A and T, again, based on the complementary strand, it will add different nucleotides. Clear? So it may keep on adding also, or it removes also. It removes, and after removing, it will add new nucleotides. Okay. Then let us see the initiation process. Actually, what happens here? First DNA A, first DNA A. It binds to the AT repeat sequence in the double-stranded DNA. Okay, let us see this. For example, first I will explain this part. Let us assume this is a prokaryotic, prokaryotic or bacterial nucleus. Okay, not nucleus. Usually, what it is called strand, double strand. Okay, so here it will choose a site where the replication should happen. Okay, so what is this site called? This is called the origin of replication. That's the reason we are calling this ORI. ORI means origin of replication. So why it is choosing only this area? And this area is having 245 base pairs, about 245 base pairs. What do you mean by 245 base pairs? Base means adenine, guanine, cytosine, right? Those are the bases. So the length, the base pair length should be 245. Okay, and it is having more AT region, A complementary T, T complementary. Okay, so here in this double-stranded DNA, okay, it will select a region where AT rich region, I mean where AT complementary nitrogen bases are there. Okay, I mean the AT should be rich here, and this is 245 base pair length. Okay, this part it will choose for the replication purpose. Got it? So here the replication starts. So where the replication is starting here? ORI region means origin of replication. Clear? So here in this OG region, in this ORI region, origin region, first DNA will binds. Okay, so what is this DNA? This is called a initiation protein. The initiation happenings with the DNA only. Okay, initiation protein binds to the origin of the replication and denatures the double strand, forming two strands of DNA. Got it? So this DNA here, it I mean it is binds to the origin place and it will denature. What do you mean by denature? It's a double stranded, right? Then it will denature so that the two strands will be unlinked or unbinded like this. Here there is a linkage, right? Double hydrogen bonds and triple hydrogen bonds between the adenine, thymine, guanine, cytosine, right? So because of this DNA protein action, this got denatured, and it forms like a bubble. Why bubble? Because there is no connection between the two strands, right? Means they got those complementary base pairs got released, means the hydrogen bond breaked, right? So this is called the replication bubble. Okay, so the separation of the two strands is happening, right? So this is called the replication bubble. So in the replication bubble, you can see there are two strands, right? Now these two strands are needed for the replication so that it will give two more complementary strands, right? Then once this bubble started, okay, like this, the bubble started, okay, then two replication force will happen, the fork, okay, two replication fork will happen. The replication bubble creates two replication force going to opposite directions, okay. So I mean, from here, it will create two more strands. Here, it will create the two more strands. Okay, so this is referred to as a bidirectional replication. The replication is happening not only in one direction; it is happening bidirection also, because this is a ring, right? See, it's a ring. When it started here, the replication will be, I mean, happening here also and here also, bidirectional, right? Then it meets here. Okay, then it meets here. Okay, because these are two strands, right? So when these two strands, I mean, broken up, I mean, broken or unwinds, okay, then the complementary strands of two will be, I mean, this side it will create one and this side it will create one. Got it? So two sides the replication will happen. That's a bidirectional. Now, uh, so we are talking about this first one, right? Initiation protein. So what the initiation protein is doing? It is denaturing the double strands, separating the two strands. Later, there is one DNA helicase. Okay, we can see this is a DNA helicase. So what this DNA helicase do? The DNA helicase, it will untwist the DNA in two directions. Okay, means it will break down the hydrogen bonds between the bases. Okay, further exposing single stranded DNA. Got it. And it also recruits an enzyme called the primase. And who will, I mean, incorporate this helicase, that is a DNAB, which there is one more protein called the DNAC. That is a protein. We call this as a DNA helicase loader. DNA helicase loader. So this DNA helicase loader will load the helicase onto this double strand like this. See, first what is happening? This DNA A protein, okay, it is denaturing the double strand. And after denaturing the double strand, this helicase brought by DNA loader means this helicase loader or DNA C, it will load the helicase or it will load the DNA B. So what this DNA B do? So as this is moving, here it is splitting the hydrogen bonds and it is untwisting two strands, right? So that it's breaking the hydrogen bonds. Got it? Which one? Helicase. What the helicase do? Helicase is loaded to the fork by the helicase loader. The DNA helicase breaks the hydrogen bonds between bases and untwists the DNA. Okay, so this is how one template and two templates will be formed. As this is 3-5 and 5-3 direction, now you can see here 5-3 direction and here 3-5 direction. Okay, then after the untwisting, then the single strand DNA binding proteins, we call these SSBs, single strand DNA binding protein. So these are the binding proteins. These proteins bind only on a single strand, not on the other strand. Okay, so these are called the single strand DNA binding proteins. We're binded to one strand. Okay, so what these single stranded DNA binding proteins do? It will prevent the strands, okay, from reuniting. Got it? See, because here the helicase is cutting, right? But the forces will be there, right? Again, there is a chance of annealing both the strands together. Again, there is a chance of formation of the hydrogen bonds between the template 1, template 2. Okay, so to stop that one or to prevent the strands from reuniting or reannealing, the single strand binding proteins are binded to one strand so that it is preventing the attachment of again the two strands. Got it? So this is the single strand binding proteins. Clear? So yeah, so one is helicase. See, DNA initiation protease. it is denaturing. Then helicase. Helicase is unbinding. Okay. Then helicase loader, it is loading the helicase onto the strand. Then single stranded DNA binding proteins, they prevent the rejoining of the two strands together. Okay. Now primase. So what is the primase here? Primase means primase adds a block of nucleotides. Okay, 1 second, I will show here. Yeah. So after binding this SSB, then later here you can see on two strands the primases will be added. So this is called the RNA primer. Okay. So we call this as a DNA primase. Okay, but the name is RNA primer. This is called a RNA primer. So this RNA primer is attached to 5-3 direction here, and here also 5-3 direction only. Okay, usually 5-3 direction. Clear? So here we have two strands, right? This is one strand, the other one is the second strand. So this strand is called the leading strand. This strand is called the lagging strand. Okay, one is the leading strand, one is the lagging strand. Why leading and lagging? See, simple here, try to understand. Here we are unwinding two strands, right? Using the helicase. Okay, you're unwinding. So it is moving, right? When you are unwinding, the length of this will be bigger. Okay, I mean the length increases here. This one also the length increases. But here, the addition of the nucleotides should happen in 5-3 direction only. 5-3 direction only. Okay, so to add the nucleotides, we need this RNA primer. Okay, where in the 5-3 direction it happens, right? In the 5-3 direction happens. So that's why a primase will be linked here in at the 5 place. Okay. Now, as the the strand is extending, then it will be moving like this. This will be moving in this direction, so that the nucleotides will get added continuously here. Then a new strand will form. A new strand will be forming. That's called a continuous strand. This is called a continuous strand. Okay. So as it is untwisting, okay, then the primer will be moving like this. So now we will be having a continuous strand, right? So there is no problem here in this continuous strand, or we also call this a leading strand. But usually what happens, the primer, it will be, I mean, constructing from 5-3 direction only, right? But here, this is a 5 direction, 3, 5-3. So it is happening properly. There is no any issue. But here, see, 5 means the opposite strand here is a 3, right? So the replication should not happen from 3 to 5 direction. It happens, I mean, it should happen 5 to 3 only, right? Now, here a primer got created, or a primer got added. Okay. So when the primer added here, it will keep on adding the nucleotides in this direction only. So when when this RNA primer is moving in this direction, so here the chain elongation is happening here. I mean, it is coming like this, right? So this primer will be adding the nucleotides up to this place only, right? Then again, but here it is not happening in this direction, right? See, here it is happening in this direction. So here the leading strand is continuous one, but here this is called the lagging strand. Okay, the lagging strand, because if it happens in this direction, then no problem. But here it is happening in this direction. So only the complementary strands will form, I mean, up to here only. Then what is the complementary nucleotides for this, I mean, for these pairs? No, right? I mean, there won't be any complementary. But as the strand is, I mean, untwisting, okay, some more primers also will be added here. Can you see? Yeah, now check this diagram. DNA polymerase pushes the SSB as it adds more nucleotides. Okay, see, I mean, as the more nucleotides are adding like this, so it's a continuous one. It's a leading. Okay, DNA polymerase 3 here helps. But here, when the primer is added here, Okay, so DNA polymerase 1, it will keep on adding the nucleotides here. Then here the strand will get finished. Here the strand will get finished. And this is the, non-base, non-complementary strand. So again, what happens? One more prime or one more RNA primer will be linked here, in the other place also. Okay, in the other place also means not only one place. So this is called lagging strand, right? So the lagging strand is linked with multiple RNA primes. Okay, then here the RNA prime will be linked. Then this RNA prime, it will keep on adding the nucleotides here. Okay, then as the chain is getting elongated or untwisted, so these primes will get keep on adding. So this is one strand. Then when the RNA prime gets added here, here one more strand will happen. So this is called the discontinuous strand. Discontinuous because it is happening like in different places, right? In different places as the double strand is untwisted. Clear? So this is the continuous strand and this is the discontinuous strand because always the polymerization happens from 5'3" direction only, not in the 3 prime or 5' direction. Clear? Now see, DNA has two strands that have opposite polarity. That is 5', 3', and then 3' to 5'. DNA polymerase synthesis DNA only in this direction, 5'3" only. This makes DNA synthesis continuous on one strand and one discontinuous the other strand. Got it? Because here, when this polymerase, when this RNA prime linked here, then from here only the nucleated or multinucleated string will form or the strand will form. But here this is a continuous. Okay. So DNA polymerase 1 synthesis DNA discontinuously because DNA poly 1 runs out of the template. Here there is no template. Again, the template is forming here. Then again, this prime will be connected here and from here it has to create new one. Okay. Then DNA polymerase 2 synthesis DNA continuously because template is always available. Clear? So how to solve this problem means simple. Okay, just now we discussed, right? RNA prime means on the discontinuous strand or on the lagging strand, multiple primers will be added. Multiple RNA primers will be added. Okay, then whenever it added, the polymerization will be happening. Okay, from 5'3" direction only. When this is happening in the 5'3" direction, then this is one strand, this is one strand, like the discontinuous stranding is happening or discontinuous replication is happening here. Continuous replication is happening. So this is called a leading strand template. This is called a lagging strand template. Okay. Now, yeah, and this is untwisted, right? So there is a helicase. This helicase is untwisting the two strands, right? But there is a enzyme called the DNA gyrase. Okay. So what this DNA gyrase do? The DNA gyrase will help, okay, to reduce the tension between the two strands. Okay, means see as a replication force, the tension generated by the untwisting DNA of DNA, okay, is relaxed by the DNA gyrase. So there is a enzyme called the DNA gyrase. So it will help, okay, again not to bind these two. Okay, because there are two strands, right? Two strands are there, but always there is a what we say, there's an attraction between these two strands, right? So to maintain that stability, okay, so there is a enzyme called the DNA gyrase. Okay, it relaxes. Clear? Now then removing the primes. So here the continuous, in the leading strand, the continuous strand will form. So there is no problem here in this continuous strand, right? But here in the discontinuous strand only, RNA primes are there, right? Here there is a RNA prime. See, when you see there is a RNA prime, there is a RNA prime. Then from RNA prime, the nucleotides will get added, right? So once the replication finished, the discontinued strand is having the RNA primes in between here, 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 like that. Okay. Then we don't need these RNA primes, right? So these RNA primes should be deleted. So how these RNA primes will be deleted means there is a exonuclease, 5'3" exonuclease. Okay, DNA polymerase 1 and the lagging strand removes the RNA primer using 5'3" activity and replaces it with again new nucleotides. Right? Means it is removing the RNA primes. I mean, once the replication finished, okay, then it is removing the RNA primes. Then it will add the new nucleotides. Okay, then after removing this one, it is adding, right? It is adding. Okay, so whenever it is remove this prime, there is a space in between these two, right? Because this is one, I mean the product of one prime, the product of the other prime. There is a space in between, right? So those spaces we call it the Okazaki fragments. Okay, those spaces are called the Okazaki fragments. Clear? These spaces are called the Okazaki fragments. So later these spaces will be ligased or attached. Okay, with a enzyme called the DNA ligase. So the DNA ligase seals Okazaki fragments on the lagging strand. Clear? So what is the Okazaki fragments? After removing this primers, okay, there are some spaces. So those spaces are called the Okazaki fragments. The removal of the primer by DNA polymerase 1 generates a nick in the lagging strand where a phosphodiester bond is missing. So DNA ligase seals Okazaki fragments on the lagging strand. Okay, like this. Then it closed. Which one? The DNA ligase. Okazaki fragment sealed by the DNA ligase. Now we have two strands. One is continuous strand, so there is no any break, there is no any Okazaki fragments, nothing, because it's happening in a 5'3" direction. Then in the discontinuous strand or lagging strand, as the multiple primers are combined, then later those primers, RNA primers were removed and sealed by DNA ligase. Okay. Now two strands got created. Okay. All enzymes come together to form a mission called the replisome. Actually, I mean, all together they will work, right? So like this. So it's a leading strand. This is the lagging strand. Okay, in the lagging strand, you can see this is RNA prime, this is RNA prime. Okay, and this is the polymerase. Two polymerases are there. Okay, then this is a hexokine. Okay, then the other enzymes. Okay, so the entire thing we call this as a replisome. What's a replisome? All enzymes come together to form a machine called the replisome. Okay, together all. I mean, uh, polymerase 1, polymerase 3. Okay, then the single stranded binding proteins, then gyrase, ligase, then uh, hexokinase. So all these are considered as a replisome. Clear? Then let us see the summary of the DNA replication. So first, helicases unwind the parental double helix, right? So this is a double strand. What the helicase do? It will unwind. It will unwind. Okay. Later, single strand binding proteins stabilize the unbound parental DNA. Then SSBs, single strand binding proteins, will be added to a single strand so that it won't again make them to bind. Okay. Then later DNA polymerase. So DNA polymerase attaches here and this side also because to add the new nucleotides. The leading strand is considered continuously in the 5'3" direction by DNA polymerase. So this is a 5'3" direction. This is actual the DNA polymerase attaches here. As this is moving continuously, the nucleotides were added. Then 5'3" direction, the polymerization happens, right? So there is no any problem in this one. So this is called continuous synthesis happening in the leading strand. Then the lagging strand, this is called the lagging strand. In the lagging strand is synthesized discontinuously. Okay, because it added here, then the synthesis will be like this. And later this is again the empty template. Then again it will RNA primer got added here. Then these are added. And again here it will get the one more prime will get added. And later it will add all the nucleotides. Like this, the discontinuous strand will happen. Then once the discontinuous strand happen, then later these RNA primes will be deleted. Okay, once the RNA primes are deleted, you can see there is a space, right? So those spaces called Okazaki fragment. Okay, So these Okazaki fragments will be later ligased like this. Okay. So DNA ligase will, I mean, close the the spaces between the the discontinuous nucleotides. So after the RNA prime is replaced by DNA, okay, DNA ligase joins the Okazaki fragment to the growing strand. Okay, so this is the overall replication process in prokaryotes. Clear? So it is happening in the prokaryotes. Now let us see the DNA replication in eukaryotes. Okay, yeah, just we'll have break for 5 minutes. Okay, just 5 minutes. Don't leave. Okay, now it is 12:04. Then we'll start at 12:10. Okay, I need just 6 minutes. Yeah. Meanwhile, I will keep my mic in mute. Hello. Yeah, I'm back. Okay, so we're discussing the DNA replication in prokaryotes and eukaryotes, right? So I hope you understood the replication process in the prokaryotes, right? It's a simple process, right? Nothing complicated here. First, the helicases will unwind. Okay. Later, some proteins were linked to the strands, not to recoil again or remove the tension between these two strands, right? Then the polymerase will do the replication. In which direction? The replication happening from 5 to 3, I mean 5 prime to 3 prime direction. Okay. So one is the leading strand, the other one is the lagging strand, right? So in the lagging strand, the discontinuous strands will get created. In the leading strand, continuous strand will be happening. Okay. Then in the lagging strand, as the discontinuous strands are happening, then there will be a gap, right? So those gap will be filled later. Okay. So if there is any phosphodiester breaks are there, then those are called the Okazaki fragment. Those are also later filled with the DNA ligase. So the DNA polymerase 1 helps in the lagging strand, and the DNA polymerase 3 helps in the formation of the new strand and new leading strand. Okay. Yeah, that's it. So here you need to remember all the steps. So here there are three steps, right? One is the initiation phase. So where the initiation is happening on a site called the ORI. ORI means origin of replication. Okay. Then where is the elongation is happening? Means after initiation, means after the denaturation, after the denaturation, then the elongation will happen. Okay, because as the helices is moving, okay, the chain is untwisting. Okay, then this is called the elongation. Then what's the termination? So once the DNA strand completed, then automatically the termination will happen. So there are three phases. One is initiation, second one is the elongation, the third one is the termination. Okay, so this is the replication in the RNA. Then coming to the DNA replication in eukaryotes. So what is the main difference between the prokaryotes and eukaryotes? You know this one. Eukaryotes means it's having a lot of chromosomes. So many chromosomes are there. It's a large genome, right? So the replicate mean the replication means it's a very big one, right? So the base page should be in GBs, right? 3 GB. When you see the E. coli, it's very small one, and it's a genome also it's approximately 5 MB only. Okay, and here the replication rate also very fast in the E. coli. That is 1,000 base pairs will be added. Okay, that is the reason when you are doing the I mean, in the laboratories, when you're creating, the artificial genome synthesis or artificial, because, when you're doing molecular biology, we'll be adding a lot of primers and we'll be creating the artificial genome strands, right? DNA strands will be creating artificially in the laboratories to find out different diseases, right? So, uh, the nucleotides are available. Usually, in the market, artificial synthesis of gene. You can create the artificial synthesis of gene because the enzymes are available in the market and nucleotides are available. Okay, and even the strands also, complementary strands are available. So that we are synthesizing the artificial gene. Okay, because to find out the actual strand of a bacteria or of some cancer cell. Okay, so that is the main concept of the molecular diagnosis, right? So usually we do in the cancer hospitals, right? Lot of molecular diagnosis based on the DNA genome sequence only, right? So that's the reason we need primers. I mean, we purchase RNA primers, nucleotides. These are available in the market. Okay, so those are all the nanotechniques. Okay, fine. Okay, we'll discuss that later. Then, when you see the E. coli, the E. coli complete genome is less than 5 MB only, and replication rate also it's more than 1,000 base pairs. But here, when you take the human genome, around 3 GB, okay, and replication rate is very slow, 100 BPs per seconds. So, to solve this, what should we do? The replication should not happen in a single place. Means on a genome, in a genome or in a double-stranded DNA, the replication should happen in multiple sites, multiple sites. Got it? So the origin of the replication will happen not only in the ORI site, okay, multiple ORI sites will be there. Clear? Why? See, single chromosome present in prokaryotes, and moreover, it's a circular chromosome, chromosomes with no ends, small genome, no nucleosomes. But here coming to the eukaryotes, multiple chromosomes are there, linear chromosomes, and moreover, the rate of adding the base pairs also it's very slow. Then it also having the chromosome with ends, then large genome, nucleosome packaging also there. What do you mean nucleosome packaging? When we have the chromosomes in packed manner, it has to unwind, right? It has to unwind. It will take time, but as there is no nucleosomes, okay, then free floating DNA is available in the cell. Then there is no need of the uh, unpacking, right? There is no need of unpacking. Right? Here, eukaryotes, we need to remove the packing also. Then, yeah. So here in the, I mean, this prokaryotes, there is no ori site origin. In one side, the replication is happening, but here it's not like that. When you see the double-stranded DNA, the replication happening in multiple sites, multiple sites. Okay, from here, 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 multiple sites is the replication is happening. Then coming to the replication process in the eukaryotes. Okay, like bacterial replication, replication in eukaryotic cells also studied in three stages: initiation, elongation, and termination. Initiation, elongation, and termination. Now, let us see the enzymes and proteins and the function of different enzymes and protein. Just like in the, you compare with the prokaryotes, right? Origin recognition complex. Okay, origin recognition complex. So this is the ORI site, right? But here what is happening in the prokaryotes, there is DNA A, right? It will recognize this area because this area should have more AT. AT means 80 base pairs, right? So when it is having more 80 and also the length of this should be 45 base pairs, right? See, in the first, I think, yeah, 245 base pairs, right? It should have 80 rich and 245 base pairs. Okay. So, and which is recognizing that place? I mean, this place, which is recognizing that place, DNA A, right? So it is happening in the prokaryotes, but in eukaryotes, it's not happening. In eukaryotes, there is a complex origin recognition complex. There is a complex. Okay, there is a complex means it contains the some proteins. Okay, some other factors also there. That's called the ORC complex means origin recognition complex. This origin recognition complex will find out or recognize where the replication should happen. Okay, so identifies origin of replication. This is the difference. Then helicase. Helicase, it unwinds the DNA even in the prokaryotes, even in the eukaryotes also. Then topoisomerase. What's the topoisomerase? It will release the supercoiling. There we spoke about the gyrase. Gyrase. What it do? Actually release supercoiling because what's the topoisomerase? Here in the eukaryotes, usually it is having supercoiling, right? The DNA is supercoiled. So to relieve that supercoiling, we need the topoisomerase. But there it exists in a single, I mean, like a uncoiled, right? I mean, it is not having the supercoils. We don't need the topoisomerase in case of the uh, prokaryotes, right? But here we need the topoisomerase to relieve the supercoiling of the DNA material or chromosomes. Then single-stranded binding proteins. Single-stranded binding proteins, these single-stranded binding proteins are linked to one strand and it won't allow again the base pairing happens, right? But here the same thing, but in eukaryotes we call this as a RPA, not the single stranded binding protein. We call this RPA. Means RPA is similar to the single stranded binding protein. Okay, so it stabilizes the separated strands, and again it won't allow the strands to meet together. Then primase. What's the primase? Nothing but the RNA primer, just like in the prokaryotes. Then there in the prokaryotes, so we spoke about the DNA polymerase 1, 2, 3 like that, right? But here we are calling this as the alpha, delta, epsilon. Okay, here we have five types of the polymerase will act. Five types of DNA polymerase will act. So what are the five types of polymerase here? Alpha, beta, gamma, delta, and epsilon. Okay, so these polymerase means in eukaryotes, five polymerase will act. Then in prokaryotes, 1, 2, 3, polymerase 1, polymerase 2, polymerase 3. Right? But here 5. So out of these 5, one is completely mitochondrial DNA, okay, which will replicate the mitochondrial DNA. So forget about this one. Even the beta also, when there is any DNA problem, then it will repair, repair. So forget about the gamma and beta. Okay, here the important are only three: alpha, delta, and epsilon. Okay, so what the alpha do? It will initiate the DNA synthesis. Then polymerase delta, it will synthesize the lagging strand. Then polymerase epsilon synthesize the leading strand. You know, there are two strands, right? Lagging and leading. Leading, there is a polymerase. In the prokaryotes, that is a 3, polymerase 3, right? So just like the polymerase 3, here the polymerase epsilon work and it will synthesize the leading strand. Okay, then lagging strand, just like in the prokaryotes, polymerase 1. Okay, here the delta will is acting, okay, to synthesize the nucleotide strand. I mean, that is a lagging strand. Then there is one pCNA also available. So the pCNA is not available in that prokaryotes. What is this pCNA? It acts as a clamp. See, when the polymerase is linked, see, when the polymer, see, for example, let us assume this is a polymerase, right? So this polymerase is attached to this strand, right? Sometimes there is chance of falling this polymerase. Okay, so not to fall this polymerase, there is a FC, yeah, pCNA. There is a pCNA. So what this pCNA do? It acts as a clamp, sliding clamp, increases processivity. Okay, it increases the processing of this polymerase activity, and it also holds the polymerase as a clamp. Okay, then there is RNase FEN1. It removes the RNA primers. Okay, so in the prokaryotes, the RNA primers will be deleted by polymerase only. Okay, RNA primers, right? So those are released. But here there is a RNase. It will remove the RNA primers. Then coming to the DNA ligase. What the DNA ligase do? It joins the Okazaki fragment just like in the prokaryotes. Then there is a telomerase. What is its get the chromosome ends? So there is one separate mechanism in the eukaryotic replication. Okay, we'll be discussing this. Slowly. Then coming to the initiation process, it is just like the same in the prokaryotes replication. Initiation is the first stage where replication begins at specific sites called the origin of replication. That is a ORO site, right? ORI, origin. Okay. So see how it recognizes that origin. When it is a prokaryotes, then it will check with the 245, then more 80 sites are there. It will recognize that one. Okay. But here, how it recognizes means origin recognition scaffold. Origin recognition scaffold. We call this as the ORC, origin recognition. Okay. So this origin recognition scaffold, along with some proteins, see CD, C6, CDT1, MCM helicase complex. Got it. So what is happening here? Before replication, some proteins they form into a complex. Okay, origin recognition complex. See, ORC. This is origin recognition complex along with the CDC6, CDT1, MCM helicase complex. This is a helicase. So helicase complex, okay, helicase loader, this one, and these proteins and ORC. All this together will form into a complex that's called the pre-replicative complex. Clear? pre-replication complex or pre-replicative complex. That's called a PRC. Okay. So what this PRC do? They will recognize the origin recognition site, or they will recognize a site, okay, where the initiation, I mean, where the replication should happen. Okay, it will find out the place. How it will find means the pre-replication complex will form. So what is the pre-replication complex? The pre-replication complex contains ORC, means origin recognition complex, along with the proteins and helicase. Okay. Then later, once this pre-replication complex formed, then anyway they will, uh, see, this is the DNA strand, this is DNA strand, then they will recognize that particular location. Then after recognizing the location, then it will unwind, right? See, uh, yeah, helicase action. Helicase breaks hydrogen bonds between bases. Okay, then double stranded DNA becomes open, then replication bubble form. But the replication bubbles form in multiple sites because this pre-replication complex, it not only identified the replication place, in one place, right? So in multiple places, the bubbles will form because the replication will happen in multiple places. Clear? So this is the complex. Now, when this replication complex will form, the pre-replicative complex will form only in the S phase. You know, right? We have different phases. Okay, I think I forgot to explain this phase. See, phase means, see, this is a cell division. You know about the cell division, right? So when the active cell division is happening, there are different phases, right? G1 phase, S phase, G2 phase, M phase, like that. We have different phases. Okay. So in the S phase only, the DNA synthesis happens, right? The replication of the DNA happens only in the S phase of the cell division, even in the prokaryotes, even in the eukaryotes also. Okay. Even in prokaryotes and eukaryotes also. So that's why here I wrote the pre-RC means pre-replicative complex will be activated only during S phase to start the replication. Otherwise, they won't get activated because whenever there is a need, then only it will get activated. So it activated only in the S phase. Okay. Then the topoisomerase action, I told you. Unwinding creates torsional stress because when it is unwinding, then it usually it is having some torsional stress, I mean stress, right? So topoisomerase removes the supercoils by cutting and rejoining the DNA. Then stabilizing of single strands. So once these double strand cut by the helicase, okay, then two separate strands will join, I mean, will get formed, right? So those are stabilized by single strand binding proteins in the prokaryotes, right? But here the single strand binding proteins, we call it as a replication protein A, RPA. See, single stranded binding proteins are RPA. It stabilizes the separate strand. Okay, so once the strand is, I mean, once the double strand is uh, untwisted or broken into two strands, then this single stranded binding protein that is equal to RPA in eukaryotes, replication protein A. Then this replication protein A, it won't allow again the addition of the base pairs. Okay, then the primer will form. What is the primer? Just like in the, I mean, the prokaryotes, RNA primes, right? So RNA prime, the primer will form. Okay, so here also the same primers will form. Okay, see the initiation phase, what is happening? Origin recognition complex binds to the origin of replication. Then what is this origin recognition complex? Complex means, this is the origin recognition complex. It will find the, it will bind the replication. But this origin recognition complex is having CDC6, CDT1, and MCM helicase, right? So all this together, it will it will form into a complex, right? Pre-replication complex. Then helicase unwinds the DNA. Then replication bubble formed. Then topoisomerase removes the supercoiling tension. Then replication protein A, that is nothing but the single strand binding, okay, single strand binding protein, stabilizes the strands. Then primase synthesis RNA primers. Then DNA polymerase alpha adds short DNA nucleotides. Okay, so this happens in the initiation phase. Clear? Simple. What is the difference here? A complex will recognize the site. Okay, then later remaining are same. Then coming to the elongation. Elongation means again the same thing here. The leading strand will form and a lagging strand also will form. Okay, but there is a small difference. There is a small difference. See, uh, let us assume this is a double-stranded DNA. Double-stranded DNA. Okay, so initially there is a pCNA clamp is there. pCNA clamp. See, in the prokaryotes, directly the helicases it will I mean it will unwind, right? But here what is happening in the eukaryotes, uh, DNA polymerase. See, this is a DNA polymerase. DNA polymerase will add it here along with the RNA prime, right? DNA polymerase will add here. Okay, so along with the RNA primer. Okay, so in the prokaryotes it will add it directly. I mean it gets add directly, right? Uh, See, this is a DNA polymerase one. Okay, so yeah, when you see this DNA polymerase, then this is the RNA prime. Okay, so it got added here directly. RNA prime and DNA polymerase it got added directly here. So there is no any other proteins. So directly they got added here. But when it comes to the eukaryotes, so what is happening? Yeah, there is one pCNA. pCNA means proliferating cell nuclear antigen. Okay, proliferating cell nuclear antigen. So this is called the pCNA. Proliferating cell nuclear antigen is there. Okay, so this proliferating cell nuclear antigen, okay, will be there is one clamp loader. We call this as the RFC. So what is an RFC? RFC is replication factor. Replication factor C. So RFC loads pCNA onto DNA. See, actually there is a clamp. So what is this clamp? The clamp is called the pCNA. pCNA means proliferating cell nuclear antigen. This proliferating cell nuclear antigen will be loaded onto this DNA, double-stranded DNA, or on both the strands. Okay, on both the strands by this clamp loader. So let us assume this is a clamp. So this clamp will be loaded using this clamp loader. Okay, so this is a clamp loader. It loads the clamp onto this DNA. Okay, so what is this clamp loader? Clamp loader is called the RFC. Clamp loader is called the RFC. RFC means that's a replication factor C. What is the clamp here? The clamp is a pCNA, that is proliferating cell nuclear antigen. Okay, then what happens? This clamp loader, okay, I mean with the hydrolysis of ATP, it will make a break in the RFC. I mean in the pCNA. Sorry, this is a pCNA, right? So this is a coiled one, right? Then it makes a break. Okay, then when there is a break, then it will easily get inserted. I mean, the DNA will get easily inserted in this break, right? Because it has to coil around the DNA. Okay. Then there is a DNA polymerase. The DNA polymerase will be added to this clamp. Means this is just like a clamp, right? When we dry the cloths, we use clips, right? Clip. It's just like a clip. So this clip will hold the DNA polymerase. Got it? The DNA polymerase will be holded by this clip. So how this clamp, like the clamp will be, this is a clamp. The clamp will be loaded by the clamp loader, RFC. This is a clamp loader, then this is a clamp, and this is the DNA polymerase. All these forms into a complex. So why this clamp? Because the DNA polymerase should not fall off, or should not, I mean, because once the grip is good, then the moving will be faster. Got it? So that is the reason here in the eukaryotes, what is the first step here? The primer synthesis. Okay, the primer synthesizing. And later, what is the second step? Loading of the pCNA by this clamp loader, that is the RFC. Okay, see, RFC plus ATP opens the pCNA ring. So it is opening the pCNA ring. After opening the pCNA ring, places pCNA around DNA. So this pCNA is placed around this DNA, and later ATP hydrolysis, the pCNA closes around DNA. Okay, then DNA polymerase uh, epsilon extend the leading strand continuously because it will hold, right. After holding, then it will move in this direction, 5-3 direction it will move. Okay, 5-3 direction it will move by adding the nucleotides. The DNA synthesis will happen. Okay, now steps of lagging strand. Yeah, now I mean this is happening in the where it happens. It happens in the leading strand as well as in the lagging strand also. But in the lagging strand, as you know, the polymerization should happen 5-3 direction, not in the 3-5, right? So just like the, I mean, just like in the prokaryotes, here also the same Okazaki fragments will happen. Okay, but here DNA polymerase delta will work in the lagging strand. Then DNA polymerase epsilon will work in the leading strand. Clear? So once the synthesis of Okazaki fragments is completed on the lagging strand, okay, the RNA primer is removed by two enzymes. In prokaryotes, it is removed by I mean the what is it, polymerase only, right? But here there are two enzymes, RNase H and FENI, flap endonucleases. So these are the two enzymes. They will remove the RNA primer. Okay. Then after removing the RNA primer, anyway the gaps will form. Those gaps also again synthesized by the DNA polymerase epsilon, because as this is the lagging strand, the epsilon will work. Then a small nick that remains is finally sealed by the DNA ligase. Okay. So this is the elongation phase. I hope you understood. Okay. So till now, what is the difference you find in both recognition site, that is the ORI site. Okay. But here, the origin recognition site here will be linked with directly a complex, right? Complex. What is that complex? So that complex is called the origin recognition complex. Okay. It is having different proteins. Okay. So it is a difference in the initiation phase. And in the elongation phase, what is the difference here? In the elongation phase, there is a clamp, right? There is a clamp. The clamp is holding the DNA polymerase. Okay. So what is that clamp? There is a clamp loader, clamp, then that is initiating the polymerization process. So that is the difference. Okay. Remaining all same except the enzymes. There we use the DNA polymerase 1, 3. Here we use the DNA polymerase epsilon and delta. Okay. Now there is a small problem here in the termination process. So what is the problem in the termination process means? See, this is the DNA. This is the DNA, double stranded. Okay. Then later, this is a double stranded DNA. These two got created, lagging strand and leading strand. But But as this is the linear, as this is a linear strand, so DNA polymerase cannot add nucleotides without the 3' OH. So this is called the telomere. Okay, this is called this the newly formed eukaryotic chromosome are linear, right? And these endings are called the telomere region. These are the telomere region. So these ending may not contain the 3 OH regions. Okay, 3 OH regions. Okay, when there is no 3 OH regions, then the complementary strand will not form. Means the DNA synthesis only up to here only. There is no red. Here also the DNA synthesis from here to here. Okay, so in one phase, what is happening? We got the replication of the DNA, but not the full length. Okay, here we are missing the telomere part. When it is happening, when it happens the next generation, or again in the next division, what happens? This is already small. Then in the next division, again it won't produce little bit part. So slowly after 4 or 5 divisions, the size of the DNA will be reduced. Got it? See, DNA polymerase cannot add nucleotides without the 3OH. If this does not fix it, chromosomes will get shorter every time DNA is replicated. Okay, so what should we do here? there is a solution called the telomere replication. End of the chromosome has a specific sequence of repeats. Okay, end of the chromosome is having a specific sequence of repeats. Okay, the telomere repeat sequence in human beings is 5 prime TTAGGG. So this is a sequence. Okay, so there is a separate enzyme called the telomerase. So this telomerase will recognize this telomere repeat sequence. Okay, so what is this telomerase having? Telomerase is having protein and RNA also. It contains a polymerase and a RNA template also. Okay, then let us see here. This place is missing. Okay, means missing DNA strand in the lagging strand and any leading strand also. So this place is missing. Okay, when this place is missing, what is this part? Telomere, right? Then there is a protein called that enzyme called the telomerase. Okay, so this telomerase, let us think that this green color is called the telomerase. So this telomerase having two parts, protein, that is a polymerase, and DNA template. So the green part is called the telomerase, and this is the prime region also. Okay, so this telomerase will extend the unreplicated end. So this is a end. So it's not replicated. So it will extend the unreplicated end because it is having the complementary sites, right? So it will get complementary to the missing DNA lagging strand, right? Then when this complement, I mean, when it is having base pair with the complementary strand, then the nucleotides will get added like this. Okay, and later normal DNA polymerase will do the addition of the other strand over it like this. Got it? So telomerase extends unreplicated end. Again, telomerase extends unreplicated end. Lagging strand is completed. Okay, simple. Or else you just simply write uh, the telomerase will add the missing replicated strand. Okay, yeah. So this is the solution for the missing non-replicated part of the linear strands. Okay, yeah. So yeah, this is about the replication in both the eukaryotes and prokaryotes. Okay, so this is very important one, and definitely you will be asked to write the answers for at least short, and yeah, I think you will get a the long answer question also. So it's very important for examination point of view. Okay, so I'll be sharing this PPT as well as today's video.